Time is running out to submit your abstract for ASM Microbe 2026. This is your opportunity to share your discoveries, amplify your impact, strengthen your CV, and get your work in front of the researchers, clinicians, and innovators who are shaping the future of the field. No prior presenting experience? No problem. All you need is curiosity, commitment, and your science. Abstract submissions close January 21st at 2 p.m. Eastern. Don't miss your chance to be part of the science that moves the field forward. Visit asm.org/microbe. That's asm.org/microbe. And submit your abstract today. We'll see you in DC.
Hey there, micro friends. Welcome to another episode of Let's Talk Micro. As always, I am so happy that you're here with me, and happy 2026. I hope you enjoyed your holidays. I did, even though at the end, I got a little sick, but I'm feeling better now. But anyway, I am so happy to be back here with a new episode. And I thought, this episode, it will be good. I thought it will be good to bring a topic that maybe some of us might not be aware of it too much. Maybe we learn about it maybe like in grad school or maybe in undergrad a little bit. But this topic is metagenomics, and it's very important. And in this episode, I'm joined by Dr. Stephen Miller, who is the Chief Medical Officer of Del Valle. And in the episode, we break down metagenomics, we explain what it is, we talk about, the application of metagenomics in clinical microbiology, the challenges of bringing it into the lab. So this episode is a little bit longer, it's like over an hour, but it is great content. So I hope you enjoy it. Let's go ahead and listen to it. Hey there, micro friends. Today we're discussing a topic that I'm not sure how much you might know about. I know that we see in the field of microbiology and infectious diseases, we see many advances, and maybe by now you have heard of things. We did an episode here in the podcast about next generation sequencing. So I thought it will be a good time to maybe bring this information to you and discuss this. So with me today, I have a guest that we're going to be talking about metagenomics. I have Dr. Stephen Miller. Dr. Miller, welcome to Let's Talk Micro.
Great. Yeah, happy to be here, Luis. Thank you for having me.
Thank you for being here. I appreciate you taking the time to be here. So I think let's go ahead and start before we jump into metagenomics. Maybe you could start by telling us a little bit about yourself, like your background, what do you do, and anything else that you would like the listeners to know?
Sure. Yeah. So first of all, I'm glad to be here and talk about micro. It's a passion of mine, of course, and really a great thing that you've got going on here for the community, and glad to contribute. So I have been interested in science, all my life, and really enjoyed, science classes, doing experiments. And as I was going through and deciding about careers, I had the, sort of pre-medical requirements and requisitions, but I was always more interested in the laboratory aspect of science and actually doing experiments, learning things. And when I discovered that there were programs that allowed you to combine a MD and a PhD, I sort of jumped at the chance to become a professional student and enrolled in the Albert Einstein College of Medicine for a dual degree program there. And my work there, I came from the background of parasitology, and I was really interested in the unusual bugs and the sort of life cycles and things that would happen with these parasitic organisms that were just sort of bizarre and weird and interesting. And I ended up joining a lab working on Toxoplasma gondii and looking specifically at proteases that were involved in the process of cellular invasion by these organisms, and sort of cut my teeth on your basic molecular biology techniques. Did a lot of cloning, northern blots, southern blots, western blots, and making transgenic organisms. You could do this with Toxo, which was made it a great model organism, making knockouts and that sort of thing. So really, learning about all the basic molecular biology tools to answer interesting questions about how this organism grows, how it replicates, how it interacts with cells. And I really enjoyed that a lot as I was going through. And of course, I was also doing, the medical training, which I found also very interesting and fascinating, and an ability to help people. And when I discovered the field of pathology, I was really drawn to it because it allowed me to combine those two passions of being involved in the laboratory and you doing, what I sort of considered to be experiments, but on people, running assays. You're not doing these things in a uncontrolled fashion. They're very good tests that you're running, but it still requires a lot of that scientific know-how in order to really do it well. And so I ended up, completing my training in New York at Albert Einstein and matched at a clinical pathology residency in San Francisco. And I was really interested in doing that because I wanted to work in the laboratory of Jim McHarrow, who was one of the major parasitology researchers in the field and had really gone from morphologic descriptions of parasites and sort of looking at clinical infections to really doing cutting edge molecular biology. And at the time, we were doing some gene microarray experiments. So I moved from doing kind of individual, PCRs and cloning to looking at these broader scale tools to investigate, how gene expression worked. And I was doing this in a variety of schistosome models. And so learned a lot about, how do you do nucleic acid amplification, but also analysis over many different targets all at the same time. And so when I completed, my residency training and postdoc there, I ended up getting a job and position at the Mount Zion Hospital, where I was where I was the director of the laboratory, and I would learned about, doing hematology and blood banking and chemistry, but I was always drawn to microbiology. And when there was an opening to move full time to be a microbiology director, that's where I went. And I spent the majority of my career actually doing microbiology testing and being a director of the microbiology lab for almost 20 years at UCSF. And so I really had a great time with that, and was, much of my role there was bringing on new molecular tests. So doing PCRs, quantitative PCRs. And while I was there, I ended up, working with a number of the basic science folks who were doing really cutting edge work. At the time, it was primarily microarray based. And this was with Don Ganem and Joe DeRisi, and later Charles Chu and Michael Wilson. And we started looking at these very broad based techniques for identifying organisms. And they were, at the time, they were characterized as virus hunters. They were looking for the next virus, and in fact had found SARS-CoV-1 by its hybridization to a particular microarray, and being able to then characterize the virus based on its homology to a whole slew of animal coronavirus probes. So we, were really interested in clinical applications of this, which really fit my role as a clinical pathologist. And over the early 2000s, things really switched from microarrays to these new high throughput sequencing instruments that were being developed by a number of companies. And as it became easier and cheaper to sequence first thousands and then millions of individual DNA molecules together, the field really switched and began using this next gen sequencing or high throughput sequencing tool as the primary method to interrogate and look for particular nucleic acid sequences in a very complex biological background. And so we were finding, new viruses and interesting cases. And the idea was always, okay, well, can we take these tools and can we then apply it to actual patient diagnosis? And, this really would turn the diagnostic world on its head because usually you have to design an assay for a particular microbe. You have to know what sequence you're going for, if say it's a PCR assay, and design primers and probes to target that. But with these next gen sequencing instruments, you were able to basically look at across all the different sequence in there and find microbial sequences without even having designed primers and probes. So it was this unbiased approach to doing that, which was really exciting because you could potentially identify any organism from the sample. And so I ended up, working with these groups to basically make this technology adaptable to the clinical laboratory setting, develop the quality control aspects of that, positive, negative controls, but also metrics to show that the assay was working, go through a validation approach, and then eventually go live with clinical testing on patient samples. So I ended up doing this at UCSF for some time. And that was really kind of the genesis of, where all of this stuff had come from. And so, that's where my sort of interest in science and microbes and medical applications really all merged together in this great environment in order to develop, the tools and the clinical approach to metagenomic sequencing.
Well, thank you for that, and for for sharing your career. So interesting. Like you have done a lot in your time. So it's I appreciate it. So one of the things that we do here in like in Let's Talk Micro, right? We kind of like starting from the basics and giving some definitions that way for the audience. Maybe those that are just from students to professionals. So maybe we can start with a little bit of can you explain and you know how metagenomic next generation sequencing works, and then how is it different from other types of sequencing?
Yeah, sure. So, I'll start with sort of, the traditional sequencing approach, right? And, but many of your listeners, I'm sure, are familiar with Sanger sequencing, which is basically, taking a particular molecule of DNA and using that in order to generate the A, C, T, and G sequence that it comes from. But the key there is you have to have a single molecule and it has to be highly amplified. So you might do like PCR to amplify that molecule and then you sequence it. So that allows you to, that's a very powerful technique. It allows you to, sequence essentially any DNA strand, but you have to isolate and get that individual DNA strand in order to sequence it. The difference between that and metagenomics is that you are sequencing essentially, a very large number of individual molecules all at the same time. So you can also call these, massively parallel sequencers. And the way that you do that, and there's different approaches for different technologies, but they separate out the molecules either on the surface of particular beads or on the surface of a flow cell, and spatially separate out the molecules so that you can then sequence them individually and see what you get. So that's sort of the difference between, standard sort of Sanger sequencing and high throughput sequencing. So now you get to metagenomic sequencing. And metagenomics, of course, means many genomes. And so what you're doing in this is you're sequencing, basically all of the different, genomes and organisms that are present in any particular sample. And so the focus of metagenomics has generally been microbiology because, most samples that you take, whether it's soil or seawater or, human skin swab, most of those genomes are going to be microbial. And so, you're interested in determining what's there. So, what we're doing with metagenomics is we're basically taking all of the sequences and identifying from the sequence itself, based on a database, what are the microbes that are present. Now, you can do metagenomics a variety of ways. You can do it based on certain conserved regions. So, a very common way to do it is to use these 16S ribosomal RNA amplicons. You basically do a PCR using conserved regions for your primers, and then a variable region that is in between the primers that's amplified, but then sequenced. And you can determine by looking at the sequence relative to your database what organisms are present. And so that was the first way that metagenomics was done. What we do at Delve is actually another level of complexity, which is unbiased metagenomics. So we take the entire nucleic acid component. So we do both the DNA and RNA portions of an extraction, and for the RNA portion, of course, you have to reverse transcribe it into DNA. And the DNA portion, you can directly amplify and look and develop your sequencing libraries. You call the preparation of the sequencing material library preparation because you are then sequencing, and each of those sequence molecules you can think of as books in a library. And you're finding all of the books or microbes or organisms within your library of amplified material. So, by taking all the DNA and RNA portion, putting that into a library that has adapters that allows you to sequence it, you can then sequence, and then you don't know what region of the genome you're sequencing. So, you have to have a very large database that includes many genomes of many microbes, and as well as human, in order to identify the origin of those sequences. And you can do that by doing, very complex computational analyses to match those sequences to the database and identify the organism that it comes from. And so that's the approach that we take for Delve in order to identify those microbes that are present in a human sample.
Okay. Well, thank you for that. And one thing I like that you mentioned, Del Bio, because right, so you when on your introduction, you were telling us, your career. So you end up at UCSF, and then how does the Del Bio come into the picture?
Yeah. So, we were doing our testing. We had developed metagenomics. We had done actually some really great clinical trials looking at how that was used, and we focused on meningitis as our main indication and cerebral spinal fluid as our sample type of interest, for a few reasons. One is that, CSF tends to be sterile. And so it's not like looking at a respiratory sample or a stool sample where you have very complex microbiomes. You should really have not much there. And if you have something there, that's a problem. That's probably an infection. So it's much easier to interpret that. And it also has relatively low human content. One of the things that, is an aspect of metagenomics is that the more nucleic acids you have from derived from any organism, including the human host, is going to sort of swamp out the signal for the other organisms. And so CSF has relatively low human amount in it relative to other tissue types. And so it becomes a little bit easier to find the microbes, sort of this needle in the haystack type of approach. So we were doing this for CSF, and we had actually gone out and started, presenting at conferences and talking about this in the scientific world. And we started getting people who were interested to do testing on their own patients. Oftentimes, I would go to a conference and talk about our metagenomic work, and somebody would come up and say, "Hey, we have a patient in the ICU who's very sick. They have, encephalitis, and we think it might be an infection, but we can't find anything. Everything is negative. Can we send you a sample?" And we said, "Sure, we're going to figure this out." So we developed essentially a small reference lab within the academic lab. And that grew over the years to the point that we really, outstripped our capacity to be able to do this testing. And so Delve really came along because there was a need to spin out the metagenomic testing that we were doing at UCSF into a commercial enterprise that could really scale and do this, and also do it more quickly. At Delve, we now, one of our real accomplishments is we're now, turning around our samples in 2 days. And so that makes it much more clinically applicable. We just didn't have the staffing and the resources to do that at UCSF. So when the company was founded, I was invited to be a part of that, and I ended up accepting and being the chief medical officer at Delve. So I've moved now full time over to Delve as the chief medical officer. I still retain my academic appointment at UCSF, and it's really great to have that as a background. But I'm now, really focused on you know using the technology that was developed and applied and expanding it. We are, expanding to other sample types and indications for metagenomic testing, but also, and what really attracted me to this position was we're building out the evidence base for what does this mean to do metagenomics clinically. What is the right patient to do it on? What is how do you interpret it? And importantly, how does this affect patient outcomes and cost of treatment? And so having the ability to do that in a commercial setting where you really move quickly, both on the product development and the research sides, is really attractive. So now I am really immersed in the metagenomic field and the chief medical officer here at Delve.
Okay, and and with this approach, with metagenomics, what clinical scenarios do you think that stand to benefit the most?
Yeah, so most of the patients that get metagenomic testing are hospitalized patients. They're often sort of the sickest of the sick. They're patients that are often immunocompromised. They may have, previous tumor or immunodeficiency or on some immunosuppressive medicines. So the types of infections that you can get in these patients is very broad because you have a lot of opportunistic pathogens. And so, we've tended to do most of our testing on these patients who have been very high resource utilizers and sort of these unknown, very confusing cases where, we think that applying metagenomics, can potentially provide a diagnosis that is really life changing for these patients, and we've seen that in some of these cases.
And before I jump to my next question, that's one thing that, as you were talking, one thing that the value of networking and conferences, for all your scientists out there, it's very important, right? So like people learn about the work that you were doing, and then it ended up turning, getting samples, and then it turned to something else. So it's always encouraged to, if you can, go out there, attend conferences, and if you see something, read journals and reach out to people, and people are always willing to, to help and talk to you. So they're always an email away. That's something to take from this as well. Okay. So there was a study in Nature Medicine that highlighted the clinical utility of your platform for neurological infections. What did the study show, and what were the most surprising or impactful findings?
Yeah. So this paper that you're referring to was published in Nature Medicine, and it was essentially a compilation of 7 years of diagnostic testing that was done at UCSF using metagenomics. And so This is the largest study that's ever been published on use of metagenomics for actual patient diagnosis with nearly 5,000 samples. And we had a lot of really interesting findings. The first was how diverse the types of pathogens were that we found and that were considered to be actually causal for infection. So we had over 430 different pathogens and organisms that were identified. So the range of potential pathogens for meningitis encephalitis is quite large, and that's one thing that makes it very hard to diagnose because you can imagine only a portion of those are cultivatable. You only have PCR tests for another small portion of them, and a lot of them are very hard to diagnose, and there may not be any tests that are specifically designed to detect it. And metagenomics, the power of it is you can find these things without knowing what it is before you find it. Of course, you have to interpret the significance of it when you find it, and that's part of the clinical interpretation of that. But there's a really large diversity of that. The other major things that we found were that, compared to all other conventional diagnostic tests, and we compared this to, direct detection methods, which we defined as those methods that can directly identify a microorganism. So that includes microbial cultures. It also includes PCR and molecular diagnostic testing. And it also includes antigen testing, like cryptococcal antigen testing, for example, in CSF. But we also included things like serology, even though that's antibody-based testing. You don't directly see the organism. We compared to that, and we also included even samples that were identified from another source. So a patient with meningitis who was diagnosed by a blood culture would be included, or a brain biopsy. And even with all those comparisons, the metagenomics from CSF had over 20% higher diagnostic yield than the combination of all of those. So a single test could actually perform better, more sensitively than everything else put together, essentially. So we were very excited about that, the diagnostic possibility. And we had a very high, positive and negative predictive value showing, high sensitivity and specificity. Now, I should say no test is perfect. And metagenomics is a test like every others. And there were cases and there are cases where metagenomics may not detect an organism, whereas another test may find it. And so we always recommend that this be done in the context of a complete diagnostic workup. We certainly aren't suggesting to avoid other testing, but what you get with metagenomics is that additional diagnostic yield in order to find the organisms that are causing these infections in more cases. And now that we have a shorter turnaround time, we can actually offer this in a clinically meaningful time frame. And so by doing this analysis of really real world data over 7 years, we found some really interesting findings that we think can impact clinical practice and really positively impact patient care for these very sick patients with meningitis and encephalitis.
And for the listeners, once you after you listen to this episode, you can go to the show notes and you can find a link to the paper so you can browse it in more detail as we always do in the podcast. Okay, so Dr. Mills, one of the things, right, with bringing tests in the lab, there's always challenges. This is micro and all the other areas of the lab. There are things like sometimes, anything from space, some technical challenges, staffing, training. So what do you see as the biggest, scientific or technical challenge to broader the adoption of metagenomic NGS in clinical microbiology labs?
Yeah, no, that's a great question. We think about this a lot, and as not only myself, but other microbiology directors and hospital administrators, you have to be cost conscious, and you have to spend your healthcare dollars wisely. And so, metagenomics is still, quite expensive relative to other diagnostic tests. And so we do have to show that, it's worth it if you're going to be doing an expensive test, and you need to be making sure that you're testing the right patient in the right way. Now, in order to, more broadly do metagenomics, I think there are some technical hurdles that are being worked on, but the actual process is more complex than many other lab tests. There's a lot of steps involved in purification and library preparation. And I look at it as much the same way that PCR was 30 years ago. So PCR was being done in clinical labs, but it was, only a few that had that expertise. You had to do things very manually. You were always worried about contamination. You were running gels to find your PCR product. The advent of real-time PCR and automated PCR really allowed labs to be able to start doing this routinely because it, had closed systems. You weren't as worried about contamination. You didn't have to have specialized labor. It could be all done automated. And so that those are being developed. There are, liquid handlers that can automate the process of library preparation. So we're seeing that the sequencers are getting cheaper and easier to operate, and also providing more and more sequencing data, better sequencing data with lower error rates. So I do see a lot of these challenges as being that were being addressed. And now I think we're getting to the point that some of the challenges here are, sort of understanding the use of metagenomics. So, there aren't really guidelines on when to use metagenomics clinically. Most of the time, it's been ordered as sort of this test of last resort when everything else is negative. And we wonder if that's the right way to approach this. And if you can potentially do, a broader test, a test that can detect, all the organisms that are potentially causing an infection earlier, can you actually shorten that time to diagnosis and treat that patient better, have fewer hospital days, which would easily justify the increased cost of testing. And so I think that's, we are slowly getting better with the technology hurdles. But now I think it's time to tackle, the issues of clinical utilization and diagnostic stewardship, which I think are very important. I also, one of the very difficult and what's often kind of the secret sauce of metagenomics is the computational part, the bioinformatic analysis. And that is where you actually take these large sequence files and align them to microbial databases to see what's there. And there's a lot of nuance and expertise that's involved in developing these, but there are now commercial products and software packages that can do this much more readily. So the level of expertise that you need to have to do the testing, while still pretty high, is coming down. And as this gets better, both on the wet lab side and the dry lab side, and or the computational side, I think we will see more and more labs, insourcing and doing their own metagenomic program. I think currently most of them, have to be pretty savvy. They have to have a research program usually in order to justify it. But we will be seeing, I think, in the next, 5 to 10 years, more of this getting into more diagnostic labs.
Yeah, I agree with that. And like you mentioned cost, and before we kind of move on to the next question that I have, but just kind of wrap this up, and um, from a clinical perspective, so how do you justify the value of a test? Like they'll detect in terms of improved patient outcomes and healthcare costs.
Yeah. So it's a great question. I think it's the you know the cost and the how you allocate medical resources is one of the major questions that we have as a society today. And I think any new test that's being developed, including metagenomics, really has to show not only a clinical benefit, but a cost benefit in order to be widely adopted. And so, as I mentioned, many of the patients who get testing are very sick, and they're in the hospital, they're in the ICU. Every day in the ICU can be thousands of dollars, and very quickly adds up to hundreds of thousands. And some of these patients, million dollars or more for a hospital admission. So, if you can make that diagnosis and get the patient on the right treatment, you can actually save quite a lot of healthcare dollars by doing that. And I think, you know, with your audience, being included to microbiology and diagnostics, I think they all understand this, that, knowing what you're treating makes you more effective in treating that, and not only gives you better outcomes for the patient, but also lowers cost. And so that's the overall argument. And we can point to particular cases that that's the case. And in fact, one of the seminal cases that really kind of really spearheaded sort of the clinical development of metagenomics was a case with a pediatric patient who was immunosuppressed and had several hospital admissions for a recurrent meningoencephalitis. And they were unable to diagnose. They were treated empirically over and over again, and they kept, sort of getting better, getting worse, coming back. When we finally were able to get a sample from this patient and do metagenomics, we actually diagnosed leptospirosis. And those of you who are familiar with leptospirosis know that it's a bacteria, but it's not something that you would grow in a culture. And you have to either have a serologic test to identify the antibody, or more commonly, a molecular test, such as a PCR, to identify the organism. And so those tests are not super commonly available. And there's a lot of variety of species of Leptospira. So some of the species may be less well detected by a PCR. That, again, the power of metagenomics, you detect all of these organisms. So we found that, this patient had dozens of tests. They had been in the hospital many days. And by making this diagnosis, we were able to target the therapy. And that the therapy was actually an old therapy, a penicillin, that was more effective than, the big gun drugs like carbapenems for this organism. In fact, it was inadequately treated with broader spectrum antibiotics, but the patient improved and did well on penicillin and recovered. So in that case, we not only saved that child from dying likely, but saved all of the hospital costs that would have occurred, for potentially for many months after, from making this diagnosis. And of course, we all would have wanted to do this earlier, right? In retrospect. And you see this theme across, many patient after patient, where, there are, many patients where it may not have a big impact, but then some of these patients where you have massive impact of being able to have that diagnosis that was made.
Wow, that's such a great example, and not only that, I so interesting, and then I ended up being treated with penicillin. Huh?
Yeah.
Wow, that
Yeah,
is a cool.
sometimes the old drugs are the best drugs, and I think it goes to show you targeted therapy is important. Yeah, and I thought another aspect of cost, we want to point not only to individual patients, but to uh, to populations, so we can understand how the costs are over different populations. And so we actually just completed a study that was done by Columbia University. It was presented at ID Week just recently. And they found this was a theoretical analysis of if metagenomics had been done earlier at the first LP during admission, could that have saved the time to diagnosis in this cohort of patients? And they found that by delivering a result within the 48 hours of that first LP, you could you could have reduced the number of tests by 64%. So you could have more than half of the tests probably wouldn't have had to be sent because you already had an answer with metagenomics, and it would have reduced the time to diagnosis by about 7 days. And so 7 days in the hospital is quite expensive. And so, that I think points to, and of course, we have to follow this up with actual, prospective enrollment data. But it supports the idea that, even with a test that's more expensive than other tests, you're saving on the bigger costs of days in the hospital. And that's where you can really save healthcare dollars.
Yeah, no, definitely, and yeah, like you say, healthcare stays are very expensive. I think I had a surgery once, like 2 days, and of course, with insurance and everything, I had to pay very little, but yeah, it was like over $60,000. So yeah, it's definitely very high cost. So okay, so you mentioned that, right? So when you started presenting your research, and people approach you, so when clinicians, they approach you, what's like the question that you get most often, from them regarding, metagenomic NGS?
Yeah, we get a lot of different questions. Some clinicians have heard about it, but don't really, know well how it works. They want to talk about that. I think more and more people are familiar with metagenomics and they sort of understand, the technology. The question that's probably most common is, when should I do it? Is my patient an appropriate candidate for metagenomics? And I think it's a good question because, we as, physicians and providers need to be good stewards of, those healthcare dollars and make sure that we're doing this test and all tests appropriately. So, they want to know, okay, what do you think that this is a good candidate? Is there a reasonable chance that this will pick up an infection? And so that depends on sort of your pretest probability assessment, but also how long has this been going on. We know that in some cases, periods of viremia can be short, and so it can be hard to diagnose cases later on. We usually do like to test them earlier. If the patient is immunocompromised, though, that's all bets are out the window, and they could have a very prolonged course with a lot of things. But they also want to know, how do I deal with detection of organisms that may not be clinically relevant. And that's a challenge for the field because, we not only find things that are, infecting pathogens, but any organism that's present in the sample. And it could be from the patient, it could be, incidental. There are many viruses, and human herpes viruses are classic, that are latent. You find them in white cells. And if you have them in a sample, metagenomics will detect them. You have to interpret that and decide, do I think that's clinically significant or not? And that's not even to mention the possibility of contamination. You can get contamination from labware, reagents, skin flora. We're all used to seeing, blood culture contamination rates, and we know that that happens. And it happens in metagenomics samples as well. That there are, skin flora, oral flora, environmental flora get into the sample, and we'll detect that. So, you have to not only be able to identify those organisms, but you have to be able to interpret that significance. We attempt to do that at Delve by providing an interpretive report that actually gives some clinical context as far as the type of organism that was detected and the likelihood that it is an actual pathogen or not, which we can know based on our understanding of what are typical human pathogens, but also what's the context in which we're seeing this organism. And we have found that, when you have a contaminated sample, you often see what we call multiple bacterial genera, or many different organisms that are present because most sources of contamination are mixed. Um, mixed organisms of a whole variety of different species. But most infections are not mixed infections. There are some exceptions to that rule, such as abscesses and the like. But you know, if you have a clinically significant infection, you know, the majority of those are monomicrobial. And so you can use the context of the result, but then ultimately you have to make that, interpretation, understanding the clinical picture. And is that organism consistent with that being an infection in terms of the patient presentation and signs and symptoms or not? And that's where, being able to understand what's going on and discuss these results is important. And that's something that we think is very important, is to keep those lines of communication open and offer those clinical consultations.
Yeah, definitely very important. And one thing we see as with different tests and one in technology and when it comes out that sometimes some might be they're meant to they come in as maybe being complementary or as a like as a supporting tool. So as far as metagenomics in the future of clinical practice, do you think that it will stay like a tool for specific difficult cases, or you think it might become like a first line diagnostic test for like a wider range of infections?
Yeah, I think that's an interesting question and something that we've thought a lot about in terms of, how what are the clinical indications for testing, So in as I mentioned, most of the cases that we started out were these patients who had already done tons of other testing and they were sort of this last resort test. But we're seeing more and more earlier use of metagenomics. And, you have to provide the evidence that this is going to be useful, but I think we're seeing that because of the decreased time to diagnosis and the fact that we are now in the commercial lab doing this with a quicker turnaround time, getting results in 2 days, whereas before it was, more than a week on average to get those results. So as you get this earlier test result, it's going to be applicable in more patients, and you are you are going to be, doing that testing earlier. And so I do think that, we're moving from sort of this last line of defense testing to earlier. I wouldn't yet call it probably first line. It's probably, second line after some initial, cultures, and now there's a whole variety of PCRs that are available that can be done rapidly and in-house. You want to do those first. But if the patient is still undiagnosed and, sick enough to be in the hospital, then you have a good argument that, that's a candidate for doing metagenomics, because, getting that diagnosis is going to allow you to get them on the right treatment. And you would rather get that done sooner rather than later, both for the patient and for the costs. And so we're now moving in that direction, and we are also developing study designs to answer this very question of, what is the appropriate time frame to be doing metagenomic testing on these patients.
Okay, and when it comes to diagnostic stewardship, how do you see metagenomics contributing to it?
Yeah, no, diagnostic stewardship is really important. I mean, you have to make sure that you're doing your tests on the right patients with the right sample at the right time. And I think the principles of diagnostic stewardship apply to metagenomics just like they apply to all other lab testing. So where we want to go with this is really to understand, what is the added benefit of metagenomics, in addition to conventional testing, because you still need to do your cultures, your PCR testing, your serologic testing. None of that is necessarily going away, but there's an added value to doing metagenomics when those aren't able to give you the answer. And so, by selecting those patients, and I think now we're looking at this more on the patient side, So it's those patients who are immunocompromised, who have extended hospital courses. We're seeing an interesting use case of patients with risk factors for vector-borne diseases, such as arboviruses and tick-borne viruses. Those are hard to diagnose, and they're a good use case for metagenomics because we can find, any any of those organisms. So, I think you have particular clinical use cases that have now a basis of evidence that you actually benefit from metagenomics, and that's expanding to become more than just your sickest of the sick patients. It's patients who might be less sick earlier in their course, and still can derive a lot of benefit from metagenomics.
Okay, and I know, like, you touch on, you talked about, test results, and right, so we get this information, and how to interpret it. Um, so when it comes to, how does your team ensure the correct interpretation of test results, especially in complex cases?
Yeah, that's been really a critical part of using this technology, and getting people, and providers used to being able to interpret these results. So the way that we've approached this, both at UCSF and Delve, is to offer what we call a clinical microbial sequencing board. And this is essentially modeled after your tumor board model, where you get the relevant providers into the same room. We're often doing this on a Zoom call or a remote discussion, but you get the primary care team, you get the infectious disease provider, you get the neurologist, maybe you have the ICU critical care person, you discuss the case. And we found this to be really useful because by having all these different perspectives, you can put the findings into the appropriate context and really make a decision, and a rational, scientific decision on what do you think this organism is actually, causing this infection? Should it be treated? What should it be treated with? Are there additional, follow-up testing that should be done to clarify and to confirm diagnosis? So we offer these clinical microbial sequencing boards really on any case, whether it's positive or negative, in order to really put the results into clinical context. And we found that providers are really appreciative of having that access and being able to talk to somebody who really understands the laboratory side of things, and can then you apply that to the patient. And I can give you a an example of a case that we that was presented at the last ASM microbe meeting earlier this year, where we detected an unusual Moraxella species in a patient as a reference lab. Yeah, you don't necessarily have any clinical context to that patient sample. When we held our CMSB consult discussion, we found that patient was a child who had a shunt infection, and in fact, that gram stain had been done from the shunt, but it was culture negative. The patient had already been on empiric antibiotics. But that gram stain showed intracellular gram-negative diplococci, which was consistent with our finding of Moraxella. And so we were able to use all of that information to really get high confidence that this was actually what's causing that patient's infection. That enabled them to decide, do we switch antibiotics? What do we do about the shunt? Do we need to remove it? And really make those decisions in a very rational way that improves that overall patient outcome. So I think because of the complexity of metagenomics, I think it's still important that we offer these clinical consults, and that we discuss the cases, because you know this is getting towards precision medicine, where we're really we treat those individual patients, and we need to use all the data that we get and put it all together in order to really, decide what to do, both for making the diagnosis, but then how do you treat that patient?
And you already you mentioned, you talked about the kind of like the role of metagenomics, like talk about the future of the clinical practice. So when it comes to it, integrating with other emerging technologies, to give a more complete diagnostic picture, how do you see that for metagenomics?
Yeah, I think this is a really cool area where medicine is, evolving and using the scientific tools that we've developed to enhance, our diagnostic capabilities. So I've really focused our discussion on metagenomics for pathogen detection and seeing, okay, what microbe is there. But there's actually a lot more data that we generate with these tests that can be also analyzed and can give insights into what's going on. And so, one of the things that that we get is from the RNA portion of the library. We actually see transcripts and genes that are expressed by the human and the human host. And you can actually take these genes, count them up, and develop models for host transcriptomic analysis that then look to see, does this transcriptomic profile look like a patient who has an infection? Does it look like a patient who has a virus infection, a bacteria infection, a fungal or parasite? Or does it maybe look like an autoimmune type of response? And that can help you further classify these patients as being okay, that's, confidently, I think this patient is infectious. Or maybe more confidently, I think this is not an infection. Maybe it's an autoimmune condition. That is actually a very common diagnostic quandary for clinicians, especially in the in neurology, where you say, well, if I have a patient with an infection, I need to treat them with antimicrobials and antibiotics and antivirals. But if I have a patient whose disease is an autoimmune condition, I want to treat with them with immunosuppressants and steroids. And that will, tamp down their immune response. But if they have an infection, that's going to be bad for them. So, I want to be sure one way or the other. And so you can use the host transcriptome data to inform that response. And so we're getting to this era of, you can call this omics medicine, where you're using very large data sets to be able to figure out what's going on. And I think that's a very exciting, aspect of this, that we can then discover new things like these host transcriptome profiles that can further help diagnostics. And so that's one of the things that we have on our research program, and we want to develop that for clinical diagnostics. And I think that's it's there's a lot involved in there, but it's going to be a very exciting journey.
Yeah, and speaking of of research, are there any like any upcoming trials or any other research that you're involved with that that will further the validity of the utility of and metagenomics in new disease areas?
Yeah, so we have some trials that are, being kicked off quite shortly. One of them is what we call Dimension, and it is a trial to basically take data from patients who are tested clinically and identify, what we can the test performance relative to conventional testing, but also, did this patient ultimately have an autoimmune diagnosis or an infectious diagnosis, and help to develop those host transcriptomic profiles as well. We have another trial that we're really excited about called Impact, that is designed to basically perform metagenomic testing on the first LP when that patient presents to the hospital with suspected infectious meningoencephalitis. And so, we talked already about, the shift toward earlier testing and really, trying to get that answer more quickly. And can you actually save time in the hospital? And with this trial, we intend to, generate some really, really high quality prospective data showing that, in fact, by doing this earlier testing with metagenomics, you can actually shorten the time to diagnosis. You can increase the diagnostic yield. You can get those patients, diagnosed more quickly, which then can lead to improved treatment, time to appropriate therapy, and ultimately, we hope shorter lengths of stay. So there's a lot of really interesting stuff that's upcoming, and I think the field also, there are many trials that are coming out showing utility of metagenomics in a variety of patient populations. We're seeing more and more publications coming out, and it's really exciting to see.
Yeah, definitely. And when it comes to public health, and metagenomics, you think about like emerging infectious diseases, novel pathogens. So how do you see, metagenomic testing playing in public health? Like what role do you see it?
Yeah, it's becoming more important in public health because as a tool, you can use it to detect, new organisms and investigate outbreaks and basically find that organism more quickly. And in fact, this was actually shown with COVID. The detection of SARS-CoV-2 and the identification was first done with metagenomic techniques. And we were able to actually, recover the genome, figure out the genome sequence, and develop PCR, primer sequences very quickly. And so we actually are seeing, good utility of metagenomics both for outbreak detection, but now for general public health surveillance. And we have a number of cases where we have identified clinical infections that have led to public health investigations, um, and have, really helped to prevent further spread of infection. So one example of this that has actually been published was the identification of these of a fungal infection, of fusarium that was associated with patients who went to a particular clinic and got spinal anesthesia in Mexico. And there was actually contamination of the surgical instruments that occurred there, and what happened was these patients became sick, and they had this unusual type of fungal infection from fusarium. We were able to identify that with metagenomics, which really led to, not only that clinic closing and fixing that issue, but also follow up of all of the patients who had gone to that clinic who were at risk of having this infection. And of course, if you get it, it can be very severe. So there were recommendations that CDC had to do follow-up testing on those patients, and one of the methods that they recommended was to do metagenomics on the spinal fluid for these patients. So you are seeing a lot of applications because of the broad ability to detect different organisms that make metagenomics really amenable to public health monitoring. You're seeing, I don't think we have time to talk about it, but food safety monitoring is another area where metagenomics is really starting to be much more much more active. And so I think by being able to really broadly, assess what's the microbial content in, not only patient samples, but, the environment, food, wastewater, you can really, use this as a tool to further public health.
Well, this has been, and I hope that the audience enjoys learning about this. I most certainly have as well. And I think I do have one question to kind of just wrap things on a lighter note. I always love hearing the responses of the guests, and we all tend to have like that, that organism, that bug that we like for any reason. It's either has like a significance. Maybe we work with it in school. Maybe it's a mechanism, maybe something visual, many things out there, maybe something with antibiotics. We all tend to have that favorite bug, or, maybe one that we dislike the most. That's also another way to go. So, Dr. Miller, what is your favorite bug?
Yeah, I still have a soft spot for parasites because I just think they're so cool, they're interesting, and they have all these really weird life cycles. And so, one of them that I find really interesting are the schistosomes and Schistosoma mancini. They live in an intermediate snail host, and then they have this schistosomula is grows and then produces these cercariae, which are these kind of free swimming little darts that are going around in the water, and they can penetrate the skin. When they penetrate the skin, they go into the vasculature and they live in the blood vessels. This particular species lives in the sort of intestinal hepatic venules. And what I find really interesting about them is, they they form these pairs with a male worm and a female worm, and the male sort of envelops that female worm, and it produces large numbers of eggs, and those eggs form granulomas, and they eventually because of this granulomatous inflammation that it produces, they transit across the intestinal epithelium into the gut, and the eggs then go into the stool and then into the water supply where they reinfect the snails, right? So this really weird kind of convoluted life cycle. But what I find fascinating about this is not only that, but these worms are very long lived. They can live for decades in human hosts. So you can have an exposure that happened, 20, 30, 40 years ago and still an infection with these organisms. And so, to me, that's just, one of the really weird, interesting examples of parasites that has so many bizarre things about it that is fascinating. And I think that's where we really, we learn, we figure out what's going on, and there's always something new to discover and to find out. So that's you know that's where I'll give you the example of a bug that I really like.
That is a great one, actually. Yeah, I heard different answers and I really enjoy that one. So thank you for sharing that with the audience. And as I said before, check out the show notes as you're listening to this. And there's links, you can find the article that we talked about in this episode. Well, Dr. Miller, this has been such a great, episode, so informative, and thank you so much. As I said in the beginning, this is something that maybe sometimes in the clinical lab, and depending on what setting you are, some people might not be too know too much about this. So I think it was very important to bring this to the audience to learn more. So thank you so much for taking the time to be in Let's Talk Micro.
Great. Thanks for having me, Luis. It's been a pleasure.
My pleasure. Well, there you have it. As always, I hope you enjoyed this episode, learning about metagenomics and how it is used in clinical microbiology. Very important. We see things a lot of, we hear things like sequencing and PCR. So it's good to learn about this. So I hope you enjoyed it. So as always, you can find Let's Talk Micro on any podcast platform. So if you go to Apple, Spotify, Amazon Music, Good Pods, we are right there. So please subscribe to the podcast, download episodes, and if the app allows you to do so, please leave a review. Thank you so much for the support. And speaking of support, if you want to do that in any way, you can do so via Venmo or via Buy Me a Coffee, KOFI. Both links are available on the show notes. And if you want to find us on socials, we are there, Instagram, TikTok, YouTube, as Let's Talk Micro, X, Let's Talk Micro 1, LinkedIn as Luis Plaza, Blue Sky Social as Let's Talk Micro. There's the website, letstalkmicro.com, where you can find information like episode updates, information about licensing, study tools, and more. So I invite you to follow us on socials, subscribe to the podcast, subscribe to the website. And if you have any questions and you want to ask them via email, you can do so at letstalkmicro@outlook.com. And if you want to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book, available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. And as always, these are some great resources from Dr. Timothy Gauthier, a very passionate pharmacist. And as you know, the Let's Talk Micro podcast is supported by the American Society for Microbiology. To learn more about microbes, visit asm.org. And that, my dear audience, it's the end of this episode. I hope you enjoy learning about metagenomics. As always, I enjoy sharing this information with you. So please continue bringing that passion to what you do. It's so important. You do such a great job. And thank you for the support. I'm so grateful for you downloading episodes, checking the content. And, I am so happy to be back here in 2026 doing more episodes. So thank you for everything. And stay tuned. Great things coming your way. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.